So our first presenter, Eliza from Uganda. The presentation titled Application Machine Learning for Selection of Site Responses in Peanut Breeding. And our presenter is Izana Chapa from Uganda.
Thank you, Dr. Kemright, for that kind introduction. I'm Chapu Ivan from Makerere, PhD student at Makerere University. I'll be presenting on application of regression and classification models for selection for leaf spot resistant in peanut breeding. Okay. Leaf spot is an important disease in peanut production. In Africa, it's the second most important disease after granite rosette. It causes up to about 50% yield losses when it occurs alone. When it occurs together with other leaf spot, it causes up to 70% yield loss. And when it occurs together with granite rosette disease, it causes up to 100% yield loss. So leaf spot is characterized by black lesions on the lower surface of the leaf, and under high severity, the leaflets become chlorotic and necrotic, and there's premature senescence and leaf drop from the plant. So several methods have been used to control the disease, including use of fungicides. Fungicides are used to control the disease. However, this is expensive for small-scale farmers, and development of disease-resistant varieties is the most cost-effective means of maintaining yield stability among the small-scale farmers. So plant breeding involves evaluation of large breeding populations across multiple breeding environments, and phenotyping of these populations involves use of traditional methods, which involve use of the visual scores. The visual scores commonly used, we have the 1 to 9 scale by Subramaniam et al. 1995, the 1 to 10 Florida scale, Chiteka 1988. These traditional phenotyping methods are subjective, labor intensive, and time consuming, and lack repeatability. And yet for plant breeding, we need the most accurate data for our selections. Remote sensing tools have been adopted as alternatives to these visual scores because these tools are fast, accurate, have high repeatability, and cheaper for breeding programs in the long run. These remote sensing tools generate large amounts of multidimensional data, which cannot be adequately handled by simple statistical methods. These simple statistical methods are not well equipped to handle complex data sets, and therefore, machine learning algorithms might be better equipped to handle this kind of data. So machine algorithms can adapt to nonlinear data and derive sensible patterns out of this nonlinear data. Some of the machine learning algorithms, like the random forest, support vector machine, the k-nearest neighbors, and artificial neural networks have been suggested for use for selection for disease severity. So these biotic and abiotic stresses often express similar symptoms like leaf wilting, leaf defoliation, and senescence, and therefore there is need to optimize the algorithms to target the particular signs and symptoms of a particular disease. And in this case, we are looking at the leaf spot. So our objective here is, one, to identify the best models to be used by breeders during LLS screening. Two, to compare regression and classification machine learning algorithms for accuracy and usability in breeding programs. So we hypothesize that the application of machine learning algorithms has the potential to improve the accuracy of selection for LLS resistance in peanut breeding. So in this, we had a total of 223 genotypes from the African collection. These genotypes were planted in 2 different countries, in Ghana and in Uganda. So we had a total of 200 in each country, planted in an alpha lattice design, and data was collected across the growing period. We scored for leaf spot using the 1 to 9 scale. disease GRD was collected as well on a 1 to 5 scale. So for our remote sensing tools, we used simple handheld cameras, an RGB camera, a Sony Alpha 600 for taking the pictures of the plots. And we also used a handheld green seeker for taking the NDVI of the plots. So after the images were taken, we used a simple open source software, that is the BreedPix from CIMMYT, to derive color space models from which included the hue, saturation, greener area, crop senescence index, and greener area. So since let leaf spot affects the coloring of the plant, we thought it wise to use color space models, which also show the color of the plant in the long run. So we right here we have images from the BreedPix before processing, after processing. So this is the green area and this is the greener area. So for analysis, we went with two approaches. One, we treated the disease ratings as continuous variables. That's where we did the regression, and we also classified the disease cause in two different categories, and we applied a classification. So for the first regression, we were able to use the correct package. We divided the data into two parts. 70% of the data was used for training and 30% was used for testing. So we used the random forest support vector machine, k nearest neighbors, the ANN, and we also used the simple linear regression and the partial least squares regression. So after these models were developed, we used 30% of the testing data set to test the models which were developed, and the accuracy of the models was based on the root mean square error and the R squared of the models. So the models with the lowest root mean square error were considered. So for classification, we went about it in three different ways. One, according to the breeders always select for genotypes up with a score of up to 5. So we decided to say if we classify all the genotypes from which have been scored on a scale of 1 to 9, we divide them into 2. So from 1 up to 5, we said breeders will accept that, and then from 6 up to 7, non-acceptable. So let's say that will be a binary classification. We shall run binary classification models for that. Then we went ahead and said, what if the breeder is to increase the number of classes along that scale? So from 1 to 3, breeder will consider it resistant. From 4 up to 6, the breeder will consider it tolerant. Then from 7 up to 9, the breeder will consider that susceptible, and those will not be selected. Then we said, okay, what if the breeder goes ahead and divides that scale further into 4 different parts? So 1 to 2, highly resistant, 3 to 4, resistant, 4 to 6, tolerant, and 7 to 9, susceptible. So the breeder might only take those which are highly resistant, and the rest will be left out. So we followed the same approach, divided the data into 2 parts. We had the 70% used for training using the correct package in R. And we had several models, the random forest, support vector machine as well. And we added the naive Bayes as well among the models. So for the accuracy, the accuracy was based on the total number of plots which are correctly classified out of the total number of plots in that set. So from the regression results, we found out that the artificial neural network, the support vector machine, and the random forest were the best performing algorithms for predicting the severity of let leaf spot. They had the highest R squared and the lowest root mean square error. So compared to the simple linear regressions, which are the ordinary statistical methods, the artificial neural network had a root mean square error of 21%. So when you compare that to simple linear regression, which had an error of 33%. So you find that there is an improvement in using the artificial neural network in predicting the disease severity. So, so in this case, we would recommend using the machine learning algorithms if you want to get the best use of the data we've collected. So going, it's not moving. The slides are not moving. Oh my goodness. Okay, so once we go to the binary classification, we find that still the random forest, artificial neural network, and the k-nearest neighbors had the highest accuracy. So these were able to clearly distinguish between the resistant and the non-resistant. So what the breeder would say is resistant and what he would not select. So the binary classification was able to accurately distinguish between the two. So when we moved on to multi-class classification, this is where we are now assuming the breeder has increased the number of classes. So we realize that by increasing the number of classes first, the accuracy of the models reduces because there are more boundaries to be made, and there's close association between the scores, there's high similarity between the disease scores, and however much the accuracy reduces with the increase of classes, it is better to have more classes because it is more practical for plant breeding. For example, a breeder would want to reduce the number of the genotypes he has selected as fast as possible. So when he has more classes, he's not able to do that. So take an example of this. When you have 3 classes, yes, you always have a higher accuracy, but what you're choosing as your most important, this is a very high number compared to when you have 4 classes. When you have 4 classes, you're choosing the highly resistant, which are around 232 compared to 371. So plant breeding always needs as few numbers as possible because the resources are not there to evaluate all these very large populations. And for selection purposes, the classification models are much better compared to the linear regression methods because the classification methods have a confusion matrix, which is very powerful. This confusion matrix helps us control for wrong selections because the cost of false positives in plant breeding is very high. Take an example of this. In this case, the breeder classified 89 genotypes as resistant. However, when run through the model said, no, these are not very resistant, these are tolerant. So the breeder would have chosen everything considering it to be resistant, yet some of them are just tolerant. Worst case scenario, we have 9 genotypes here which are susceptible, and yet the breeder has scored them to be resistant. So this is very expensive for plant breeding case. So in conclusion, machine learning algorithms can improve the accuracy of selection for resistance. Artificial neural network is recommended. The support vector machine is recommended as well. And the random forest is probably the best overall. So classification models are more practical for selection compared with regression models. So I would encourage people to adopt the classification models. And with more classification classes, the accuracy of the models reduces, but improves the precision of selection for the desired trait. In this case, let's put resistance. For our future work, we intend to use the drones rather than the handheld devices because we shall be able to collect more data, higher frequency of data collection, and we intend to subject this data to deep learning models so that we develop more robust methods for detection of the disease, detection and selection of the disease in real time. I would like to thank my supervisors, Dr. Maria Balota, Dr. David Okelo Kalule, and Dr. Cyrus Ongum, and Dr. Richard Oteng Frimpong, Abila Chandel, and Emmanuel CA for all the help. Thank you very much. If at all there are any questions.
Thank you very much. Especially thank you for your efforts coming from Uganda. So do we have questions for our speaker? If you'll get them to me, I will repeat them to him. One question is, as far as primary breeding efforts, for those of us who don't know about breeding efforts in Uganda or Senegal, what is the primary breeding? Is it disease resistance, drought resistance, and how important? And I guess that's all tied into yield. So in your breeding programs, what's the most important efforts or facets of it? Is it disease, drought, or all things included?
Yes. Let me speak for the case of Uganda. So for Uganda, there is just a minimum for any variety to be passed. One, it has to have granite rosette resistance in the background. It has to have late leaf spot resistance in the background. Then for some areas, we need drought tolerance as well. But overall, at the end of it all, the yield is the most important thing. But the disease resistance is non-negotiable. It has to be in the background of every variety released.
Thank you very much. So not so different from the Southeast, where we have to have tomato spotted wilt virus resistance. So great. Any other questions? One more question, or one last question from Dr. Bertioli.
This program being able to also act as
The question is, do you also see this program being able to look at resistance to GRV?
Yes. We have work which is ongoing right now, and there is someone who is currently handling that. There is Esther Achola is currently handling that, and yeah, I believe she's going to have for us results very soon. There's work ongoing on that.
Great. That's the time we have for questions. So thank you very much, and thank you for participating in the JOSUG competition this year, sir.
Oh, thank you very much.
One thing to note is while we do not generally allow Zoom presentations, we feel that being in person in this meeting one of the critical components, but we make an exception for students, graduate students, because they do not have the means to absolutely want to engage. Why we have graduate student presentations from international locations, we do not have absolutely want to encourage graduate student participation wherever. So, our next presentation is entitled Conversion of Early Maturing Spanish Diabetic Variety Resistance Early Leaf Spot Using an Arachis Cardentii Derivative Line as a Donor. Joseph Gomez is our presenter today from Senegal.
Joseph, you can begin.
Okay, do you hear me well?
Yes, we can.
Okay. Thank you, Dr. Hemi. Hello, everyone. I'm Joseph Gomez from Sheikh Anta Diop University, and I'm currently doing my PhD activities at Serras Laboratory. I would like to thank the APROS for this opportunity given to me to share some of our results in genetic improvement of cultivated peanuts resistance to early leaf spot using wild species. This work was done in partnership with the Bertolli's team, who kindly provide us with wild peanut seed. Well, early leaf spot is one of the most widespread and devastating peanut diseases in the world. Uh, the causal agent, Cercospora arachidicola, cause necrotic lesion on leaf, which may lead in severe defoliation and yield losses up to 50%. In Senegal, the area called Peanut Basin and the south part of the country are the most infested zone, and the risk of yield losses there is increased by the use of early maturing varieties, which are susceptible to early leaf spot, as do cultivated peanut in general. In contrast, strong resistance to a wide range of pathogens have been described in wild species, and uh, several wild species have been characterized as resistant to early leaf spot. And among them, Arachis carenaceae has been largely exploited in breeding program, initially for early leaf spot resistance, and now more importantly for late leaf spot and rust resistance. And Berkeley work in 2021 have shown that 251 peanuts line and cultivar across the 5 continents possess at least one segment of resistance from Arachis carenaceae. So in our study, we used one of the interspecific derivative line from Arachis carenaceae, CS16, which possess a resistant fragment on chromosome A2 and A3 to develop an advanced backcross population. And the main objective was to introgress CS16 resistance into the genome of the early maturing local variety, flower 11, which is susceptible to early leaf spot. So to achieve this objective, we developed a working population by crossing flower 11 and CS16, and F1 through F1 identification was made using SSR molecular marker, and the identified ones were crossed to the recurrent parent flower 11 to generate the first generation of backcross line. These individuals were in turn genotyped using SNP anchored marker to CS16 fragment on chromosome A2 and A3, and only backcross individual which display CS16 fragment on chromosome A2 or A3, or in both chromosome A2 and A3 were selected, and these BC1 was advanced by 3 consecutive backcross steps. And at each step, we repeat the same exercise, meaning genotyping with the same set of SNP marker and selection of the individual carrying CS16 fragment. So at the BC4 stage, we performed two steps of selfing, during which we did a phenotypic selection based on characteristics like plant habits, precocity of flowering, pod and seed characteristics like size and color. We did it to select the most similar line to flower 11, because we didn't apply genotypic selection for the background for flower 11 genome. So at the end, we got 7 BC4F line, which we evaluate during 2 years at Neuro Research Station for disease resistance and also for agronomic performance. For the evaluation, we assessed early leaf spot resistance using the 1 to 9 point scale method by Subrahmanyam. And disease resistance was evaluated every 10 days from the appearance of the first symptom. We also use the score obtained to calculate the area on the disease progress curves. Another parameter collected was day to 50% flowering by counting the number of days from sowing to flowering of the half of the plants on a elementary plot. We also assessed pod weight, palm weight, pod 100 pod and 100 seed weight on a post-harvest material dry. So for statistical analysis, we did an analysis of variance using the AOV models. And from these models, we exploited the Bloob's adjusted mean that we use to perform a mean test comparison between flower 11, the recurrent parent, and each of the line. At the end, we calculate an index of selection using this formula presented here, where W were for given weight and T for threats. We prioritize resistance to early spot. That's why we gave the maximum weight to IUDPC, and the rest of the weight we divided into the other threats. So as a result, we observed a significant variation between line for ELS resistance in both 2022 and 2023. And we also observed that several lines outperformed the recurrent parent, flower 11, for decisive resistance, suggesting that the fragment from CS16 effectively bring resistance to ELS to the line. Here is the summary of the analysis of variance for the rest of threads. And in general, we observed significant variation for all these threads at 5% threshold, except for pod weight in 2022. So with the LS mean obtained from the models, we performed a unit test to compare flow 11 and each line. And here is the graphical representation of the test for least spot resistance in both 2022 and 2023. On the x-axis, we have the group comparison, in other words, comparison between flower 11 and each of the line. And on the y-axis, we have the estimation of the mean difference. Significantly different groups are represented by blue intervals, while if there is no significant difference, the color is a red interval. And interval above the green line here, materializing the 0, represent individual which have higher mean than flower 11. And under the green line, we have interval representing individual which have lower mean than flower 11. As you can see, in general, in 2023, all the line outperformed flower 11 for ELS resistance because they are all significant. They have all significantly lower value of IDPC than flower 11. The same was observed in 2022, while 2 of the 7 lines didn't show significant difference with flower 11, even if they have a lower IDPC than flower 11. The same tests were performed for the other characters like Holm production, pod production, in both 2022 and 2023. And here we can see that in general, no line didn't differ significantly from flower 11, except this one. But in general, all the line were now significantly different from flower 11, even if several lines produced more Holm than flower 11, but the difference was not significant. The same was observed for 100 pod weight and 100 pod seed weight and 100 seed weight in both 2022 and 2023. And here also, in general, the line was similar in 100 pod weight and 100 seed weight to flower 11, meaning that the line that we have developed are more resistant than flower 11 for early leaf spot resistance. But in terms of productivity, these lines are similar to flower 11. So they conserve the potential of flower 11 for agronomic performance, and they are better than flower 11 for disease resistance. So we performed the index, we calculated the index of selection for each line, meaning a combination between resistance and agronomic performance. We prioritize resistance, so that, as I said earlier, we gave the most weight to resistance and the other weight for pod and seed production. And here, as a result, we observed that four of our BC4 line have shown a better index of selection than flower 11, meaning that these lines combine perfectly resistance to early leaf spot and productivity, opening the way of substitution of the susceptible line by also early maturing line, but more resistant to ELS. So the take-home message of our presentation is that we successfully transfer fragment of resistance to ELS from CS16 into flower 11 genome, and all the line we have developed were more resistant than flower 11 and are all quite performant in agronomic traits like flower 11. And we have for 4 of the 7 line show it a great index of selection compared to flower 11, meaning that this line may replace flower 11 in the infected zone by early leaf spot. So in perspective, we plan to perform a multi-environmental test of this line in the peanut basin, but also in the south, to confirm their resistance and their agronomic performance. We already did the sowing activity at Nyoro, and now currently I'm here at the south of the Senegal, here in Djibolo, to install the 2024 trial. So I would like to end my presentation by thanking all the institutions that supported this work, all the people, and thanks again the APRES for this opportunity, and thank you all for your attention. Thank you.
Thank you very much to our speaker. We have time for a couple questions. I'll start the questions off with, in my experience in the Gambia and elsewhere, late leaf spot has been more of a problem than early leaf spot has been, and I'm curious to know your experience with that. And the second part of the question, sir, is does the breeding efforts with the Cardinaceae, if you're looking for resistance to early leaf spot, does it also translate into resistance to late leaf spot?
Oh, according to my experience at Nyoro, early leaf spot is more present, even if we have traces of late leaf spot. But in my experience, I'm doing activities here since 2020, and in each year I observed that early leaf spot is more present than late leaf spot. We also use other interspecific lines, but as Arachis cardinaceae, as I said earlier in my presentation, as this word have been used in the past for early leaf spot, and I think that if I'm not wrong, several lines have been developed according to Sugg 1984 for early leaf spot. So I think that it's a good material for early leaf spot, and our results show that for flower 11, if we compare our line to flower 11, we have seen that fragment from CS Arachis cadenaceae bring us resistance for early leaf spot.
Thank you very much, and that's the time we have for questions. And again, we appreciate your participation all the way from Senegal. It makes our meeting better. Thank you, sir.
Thank you.
I'm gonna go with the title that's on here because it's easier for me to read. There's not a word to pronounce. So I'm gonna go with novel source of root knot nematode resistance for peanuts. And if you look at the program, it's got many part words, so I'm gonna go with that. And Mina Barnes is our presenter. Thank you.
Thank you, Dr. Kimmeray. Thank you all for being here. Good morning. This is a long-term project in our lab, and I'm very excited to share the most recent results. I'll briefly talk a bit about nematodes and the work that's been done so far in this problem. Is that better? Yes. Okay. Thank you all for being here. Okay. I'll briefly introduce the work that's been done so far in this project and a bit about the problem as well. Root knot nematodes are microscopic worms that live in the soil, and they can be very devastating pathogens of peanuts and many other crops. A farmer may first realize that they have a problem with nematodes if they see chlorotic, patchy areas of their fields like this, but the true extent of the damage won't be evident until you turn the crop over. At which point, you'll notice that the root system has these galls or root knots affecting it, and the pods will also be damaged and discolored and much lower in yield compared to what we expect from a healthy plant. Nematode management is also very expensive. A case study was done in Mitchell County, Georgia in 2021, where it was found that 65% of peanut acres were treated for nematodes at a cost of $35 to $105 per acre, totaling somewhere between $560,000 to $1.56 million in Mitchell County alone. Of course, nematodes don't only live in Mitchell County, Georgia. They live pretty much everywhere peanuts are grown in the United States. And if we extrapolate these numbers, US peanut growers are likely spending somewhere between $38 and $113 million in nematode prevention alone. The most cost-effective and efficient way to manage nematodes is by planting resistant cultivars. Hopefully some of you were able to attend the fantastic Charles Simpson session yesterday, and I don't have to convince you of the utility of wild species for these goals. But suffice it to say that the cultivated species has absolutely no source of strong resistance in its primary gene pool. We are completely reliant on the secondary gene pool for these sources of resistance. We do have several resistant cultivars that have been released over the years, but in fact, they all trace their resistance to exactly the same source, which is an introgression from the wild species Arachis cadenaceae. This source of resistance has served us very well for nearly 30 years, but when we rely so long on a single source of resistance to a major pathogen, it becomes an inevitability that that pathogen will overcome the resistance. So we are very interested in bringing in new sources of resistance. When we look at what happens when peanut is challenged by nematodes, the nematodes first infect the root tip and colonize it. and then move further up into the root and establish feeding and reproductive sites, which are the galls or root knots. On the other hand, the wild species Arachis stenosperma, when is challenged by root knots, the nematodes first have a hard time penetrating the root tip, and those that do then fail to establish feeding sites because a hypersensitive response initiates localized cell death around the nematode. So there's actually two forms of resistance here, and they've both been genetically mapped to a region of chromosome 2 of Arachis stenosperma. So with a location identified, work began to bring this into the cultivated species. To do so, we cross it to another wild relative and then double the chromosomes to form an allotetraploid that's sexually compatible with cultivated peanut to bring it into the primary gene pool. And from there, an F2 population was developed between the wild allotetraploid and the cultivated species. In the F2 population, resistance to nematodes was tested in greenhouse bioassays and genetically mapped. And similar to what was seen in Arachis stenosperma, we see two strong resistance QTLs on chromosomes AO2 and AO9 of these backcross lines. The resistance on chromosome 9 is a bit complicated for reasons I don't have time to go into. So the work so far has really focused on chromosome AO2 on that QTL. As I mentioned before, this has all been done in bioassays up to last year. So the primary research objective of this work was to test the stenosperma-derived nematode resistance in field conditions. We also wanted to look at yield of our backcross lines. We wanted to assess the agronomic qualities of them as well, and to begin to ask how this introgression is providing resistance. So I'll briefly go through the materials and methods we used to answer these questions. From the F2 population that was developed before, recurrent backcrossing was done with marker-assisted selection at each generation up through the BC3 F2, at which point the plants were taken to the field for a couple years and evaluated for yield and disease resistance. The most promising lines were then selected for genotyping and sorted into two groups based on the presence or absence of introgression in chromosome AO2. We took a subset of 5 AO2 positive backcross lines and 2 AO2 negative backcross lines to the field in 2023. We had 3 field sites last year in Attapulgis, Georgia, Tifton, Georgia, and Midville, Georgia. The phenotyping that was done for a root knot nematode included counts of the infective J2 stage nematodes from soil samples taken from each plot, as well as galling incidence, which is a measure of the percentage of affected roots in a plot. We also use an index that I won't present today. Some other phenotyping that was done included yield and TSWV evaluations at all 3 locations. We were able to measure leaf spot resistance in Midville. In Attapulgis and Tifton, we looked at stand emergence and seedling vigor. And in pods from Midville, we also evaluated for pod constriction using a scale I'll show later. And we looked at canopy closure data using drone imagery. I won't have time to present all of these results today, but I do plan to touch on each of the research objectives that I laid out before. And the first of those, of course, was evaluating nematode resistance in the field. When we went down to our nematode plot at the time of harvest, we were very happy to see that the susceptible cultivar Georgia 06G, which is the most common cultivar in Georgia, was showing severe damage, whereas our backcross lines with the resistant QTL were looking very healthy. When we turned these plots over, we found that between 60 to about 95% of cultivars showed nematode damage in their root systems. And we saw very similar numbers in our backcross lines without the resistance QTL. This was in stark contrast to our backcross lines with the QTL, as well as the resistant cultivar we included as a check, which showed essentially no damage. And this difference was very statistically significant according to a Duncan test. We saw a very similar pattern when looking at J2 counts from the soil samples. So our conclusion here is that the resistance from Arachis stenosperma, like that from Arachis cardinacei, is essentially total resistance. We also wanted to look at yield. And when we look at yield under nematode pressure, it's unsurprising to find that it's largely a function of resistance, with resistant lines and cultivars significantly outperforming the susceptible ones. But we were really more interested in yield without nematode pressure, because one of the main reasons that the resistant cultivars available today haven't been super widely adopted is because they have relatively low yield potential compared to the more common cultivars. So we were very happy to find that all of our backcross lines, with or without the introgression, shared a statistical grouping with the leading cultivars. And we actually had 2 lines that outperformed Georgia 06G in both conditions. So the take-home message here is really that, of course, the resistant lines outperform susceptible cultivars under nematode pressure, but actually that they can also remain competitive without nematode pressure. We also wanted to look at agronomic quality. One important agronomic trait is the 100 seed weight. Wild species, of course, have very small seeds. Cultivated peanut has much larger seeds, which is what processors expect. And you can see here that backcross lines with or without the introgression have a seed size that's comparable or even slightly larger than that of the cultivated varieties. We saw a very similar pattern with pod constriction. Wild species generally have single-seeded pods, whereas the cultivated species has double-seeded pods that are very large. And when we look at the backcross lines, we can see quite a bit of variation in pod constriction, which refers to the tightness of the pod in between the 2 seeds of a double-seeded pod. What we really want is something like the cultivated species, which is what processors and manufacturers expect. So when we plot the cultivated pods on this scale, they all score as a 3 or a 4. This is a density plot. And when we scored our backcross lines, we saw a very similar density plot with means that were not statistically significantly different. So taking these 2 results together, as well as others that I did not present today, we concluded that the agronomic traits and qualities we looked at were comparable to the cultivated species. We were able to recover these in as few as 3 backcross generations, which is likely a testimony to the success of the marker-assisted selection that was done in population development. So lastly, I wanted to look into how this introgression might be providing resistance. So I started with the publicly available sequences for cultivated Anorachis tenosperma chromosomes 2. And I did a blast search using sequences from soybean for NBS-LRR genes. These are genes that are very commonly involved in disease resistance and are likely responsible for the resistance from Arachis cardinacei. I found a good number of likely NBS-LRR genes in both species and then sought to compare them to each other, expecting to find a couple in Arachis tenosperma that are not present in the cultivated species. To my surprise, they all had a homolog in cultivated peanut. So then I moved on to more of a sequence level comparison, looking for SNPs and especially for high effect variants in these SNPs. So the sort of high effect variants that I was looking for include frameshift mutations, splice site variants, and premature stop codons. When I filtered these to only those that occur within NBS-LRR sequences within our resistance locus on chromosome 2, you can see the numbers that I found of each. I chose to focus on premature stop codons and to illustrate the effect that these can have. I want to give an example. If we consider this rectangle to represent the length of a typical NBS-LRR protein, there's the NBS site, the nucleotide binding site, and then further down in the protein, there's the LRR, or the leucine-rich repeats. These are the parts of the protein that are actually involved in binding effector molecules from pathogens and initiating a cellular response. When we look at one such NBS-LRR sequence from Arachis tenosperma, it follows this pattern very closely, and it's highly similar to its homolog in cultivated peanut, 98% similar up through the nucleotide binding site. But in the cultivated copy, there's a premature stop codon that leads to the loss of the LRR that's actually involved in recognizing and responding to root knot nematode, or to whatever it is. I actually found 7 such examples within our resistance locus. So my theory here, I don't want to call it a conclusion yet, is that introgression from Arachistinosperma may bring back functional copies of NBS LRR genes and proteins involved in recognizing and responding to root knot nematodes. Briefly, some future directions. I mentioned resistance on chromosome 9. We currently have a field trial with lines with introgression spanning chromosome 9 in a sort of stepwise fashion. Hopefully this will give us a sense of the size and position of introgression necessary for strong resistance on chromosome 9. We also know that the high oleic trait is on chromosome 9, so we're hopeful that this new source of resistance will be available in standard and high oleic lines. And our two lines that outperformed O6G are going further down into the cultivar development pipeline in intermediate yield trials conducted by Dr. Nino Brown. So that is all. Thank you to our funding sources and the University of Georgia for making this work possible. Thank you to our technical support and my lab members, and thank you for your attention. Yes.
What was the average compared to
The first cross was to runner 886, and most of the recurrent parent crosses were done to 564-610, which is a breeding line. Yes. A possible reason. Yes. I haven't had the chance yet to compare the larger seeded lines to the genotyping in detail, but Danielle Fonseca's group in Senegal has identified some wild alleles associated with larger seed size. So that may be a possibility. It may also just be noise from the number of samples that were taken, and maybe it would average out to the cultivated size with more sampling. Stephanie. I don't think Hapla is very present in the Attapulgis field. It's largely Arenaria. I don't know if the species level differentiation was done this year, but it has been done from that field before.
That's time again. Thank you very
Thank
much.
you.
I believe our fourth paper has been withdrawn, and so we will take a break until 9 o'clock. Even though we can go, we would like to go right now. Get ready for your coffee. 9 o'clock, we'll be ready to go. I don't find your seat. Can't hold this one back much longer. He is kind of loaded and ready to go. Am I right? All right. Our next presentation is titled Magic Peanut and
Pangenome.
Genome. And then the third F82 gene associated with high-oleic acid content. Peanut, Ethan Thompson, your representatives.
Thank you. Yeah, I want to make it a little bit difficult for you, Dr. Kemmerai. So,
Succeed.
everyone, I'm Ethan Thompson, and today I'll present some of my research. The main idea is that we use two different resources to understand a high oleic content within peanut. So, brief introduction, oleic content. So 50% of our dry weight in our peanut seed weight is composed of oil. Oleic acid is a major component of this, from 40 to 80%. And this oil composition is important for our peanut product at the end of the day. So some of our stuff like peanut M&Ms, they like high oleic for the longer shelf life. where low oleic is desired for our peanut butter production. So they're competing with each other for the growers and trying to get the growers to grow certain varieties of oleic. So we need to understand what kind of varieties we're growing and understand how we could use that in a breeding application. Okay, so because of that, FAD2A and FAD2B mutants have been found. They use markers in a lot of breeding programs. And so these markers are a SNP location for FAD2A, and for FAD2B, we know that there's an insertion. And these markers have been used before to pyramid high oleic trait with the nematode resistance here in this paper right here. Well, previously we've had these two genes shown, and that's what we just described from that paper. And so here in this paper by Dr. Branch, they're looking at, okay, well, we have these two recessive genes. And so where are we getting this middle oleic lines stuff coming from? We see that it's not just black and white. So initially we thought that the middle oleic was just a combination of these two recessive alleles where you only have one instead of both to give you the high oleic. So when they use this in this paper, they try to cross it to try to understand the Mendelian traits that are moving around, and unfortunately this is not the case. So they hypothesized that there are some other genes that besides this FAD2A and B that we need to look at. Okay, so that leads me into my objective to identify the distinct genes that affect oleic acid content by leveraging our two resources, Peanut Magic and Pangenome. So what are they? Peanut Magic is a multi-parental advanced generation intercross population. We took 8 parental lines and crossed them in a simple funnel-like design. So you can see how they cross all the way down into our real population of 2775. Of this 2775, that's quite large. So we took a subset of 310 reals, we call the magic core, and we utilized for the study. We sequenced these whole genomes at 1x coverage, and we phenotyped the seed for 2 years. Okay, so what is a pangenome? So we took these founders of our Peanut Magic population and we sequenced them at 25x with PacBio. So these are long reads, very accurate sequencing technology. So we assembled them and constructed them into our assemblies and then into a population-specific pangenome. And so what I mean by pangenome is we can see here the 8 different colors representing our 8 different parental lines. We can see that when they align all together in a region that is identical, they'll be the same. So it'll be represented by the single bar. And then we can have these paths that we traverse in our pangenome to allow us to represent variation. So right here is FAD2B. We know that there's an insertion in FAD2B. And so we can either have lines in this population that don't have the insertion or a functional variant, or our mutant variant, which is our insertion. So this would be represented as an indel. Okay, so we could do this across the entire genome to try to understand what variations that we have everywhere, and to remove the question of what do we have in the population. Well, now with this, we can understand truly what's happening in our population. So this creates a massive library of data that we can pull from. So you can see out here heat maps of all of our variants throughout our genome. We have our A genome and our B genome over on this side. We can see all of our different variations, our SNP variants, and our non-SNP variants. So these are some of our more complex sites. It allows us to understand the similarities between the 2 B genomes, the A and the B genome. So that's what these bars right here represent. So it's kind of complicated to understand these 2 genomes. It's to be able to tease them apart. And that's what this pangenome allows us to leverage that even more. Okay, so now since we have this library of all this data, we can understand what's happening in our population, so we can create markers to track these recombination points of our recombinant inbred lines, our reals. So we took our sequencing data, aligned it to our pangenome, and generated 463,000 markers. And this includes indel sites, so insertion and deletions, and these complex sites, which could be a little bit more tedious to deal with previously without a pangenome. So we can see that we have our densities of all variations in the populations, our markers that we can utilize in our population, and then our founder-specific markers. And so this removes our marker biases that we've had previously and improves our marker quality. We can increase how well we can see our population, and we can understand what's happening at a more finer details. we can see where these markers come from and then understand quite easily where these traits are coming from in an individual line. Okay, so quite a bit, I know. So we can, now we need to look at our oleic content to go, okay, are we seeing what we're seeing, just 2 genes or multiple genes? So with our oleic content, our seeds underwent gas chromatography mass spec to create phenotyping data over 2 years, 2021, 2022, and then the average. We can see that we have a normal distributions of normal and middle oleic lines, or low oleic and middle oleic phenotypes, and then we have a peak of high phenotypes. Okay, and see this normal distribution is suggesting that there are more than just 2 genes segregating within our population. So based on that, we know that we have some markers that we can utilize. So let's utilize these markers to genotype our population to see, are we seeing anything different than just 2 genes? So we took FAT2A and FAT2B PCR markers, used it for genotyping. And we can see here's our phenotypic data for 2021 and 2022. And for our high oleic line right here, we can see that when there's mutants, we're seeing high oleic. And that's the theory based on those 2 genes. However, we have several other lines in our population that are mutant from both FAT-A and FAT-B, but do not possess the high oleic phenotype. Okay, and so that would suggest that 2 markers are not enough to determine high oleic within populations. And so within our Magic Core population, we have 7 out of the 310s that were identified as mutant for FAT-A and FAT-B while still being middle oleic. So we want to understand what's going on. So we know that there are more than 2 genes segregating in this population. So we want to be able to track that. We could utilize GWAS, which is genome-wide association study, to compare our markers against our phenotypic distributions to identify precise locations on the genome where these genes reside. So we can see our first location right here, chromosome 9, and that lines very well with our FAT-A location. But we see a much larger peak on on chromosome 19, where we know where FAT-B is at. So we can zoom in on chromosome 19, and I've highlighted the exact location of FAT-B. We can see clearly it's right here, but there's something else upstream of FAT-B that is even more significant. So something else is going on upstream of FAT-B. Well, we have annotations online. We have peanut base. It's very easy to go to peanut base and see what genes that we have available in that location. And so when we go to peanut base, we can see upstream there's a fatty acid desaturase 2 gene identified 1.3 MB away from FAT-B. So we named it FAT-C to go along with that nomenclature, FAT-A, B, and now we've identified FAT-C. Okay, well, that's just an annotated gene. Is that in the right place at the right time? Is this functional or not? Well, we can see with the research was on peanut base, so there is transcription in our seed locations as is progressing. So this is data from Dr. Clevenger looking at the development of our peanut seeds. We can see that this gene is being expressed in the seed while it's growing and developing, which makes sense because TIF runner, so it's about 40%. So this gene is being expressed there. Okay, well, so we have the expression of this gene. We have our large library to understand variations in our population, so we can leverage that to try to understand, well, what variation do we have for FAT-C in our pangenome? We can see from previously that there's no variation within the gene itself. There's actually variation upstream of our gene, suggesting that there's a transcriptional inhibition from changing this promoter region. So that leads in the question, we have 2 variations right here. We can blow it up and look at it through the pangenome. We can see that there's an indel, which is an insertion deletion. So that'd be an insertion here, deletion there, or a SNP, a single nucleotide polymorphism. So, well, what's the cause? Is it a SNP or an indel? Because we need to know whenever we're trying to give this to our growers and our breeders to be able to track this in a population to really get down to the causality. So I made a pipeline to target these sites of interest. And so I generated all this data using my sequencing data, my pangenome, to try to genotype these specific sites, the SNP that we saw in our indel location. And we can see with our high oleic line right here, that has mutant for FAT-A and FAT-B, in the mutant SNP location while having the wild type indel location, meaning that the indel is not important for FAD2C. However, it seems that the SNP is important. So we have, when you have a mutant FAD-A and FAD-B with a wild type SNP location, we can see that in these 3 representative samples right here, that we're now no longer maintaining high oleic when we have the SNP on our FAD-C promoter region. So the mutant SNP seems to give off our high oleic. So in conclusion, we demonstrated that there are more than 2 genes controlling high oleic in peanut. We've identified a 3rd gene, FAD2C, that's neighboring FAD-B, and this proximity explains the difficulty in isolating the 3rd gene for several years. And this also explains the increased significance that we see in previous literature of FAD-B being much more significant than FAD-A. And finally, we linked the SNP variant in the FAD-C promoter region to high oleic. So in the future, we'd like to apply this FAD-C calling in breeding populations to be able to use that and leverage that to have accurate phenotyping when we're trying to have breeding populations for high oleic cultivars. So finally, I'd like to acknowledge my committee committee members of my lab, and some other people that have helped me along the way. With that, I'd love to take any questions that you guys have. Yes. So based on the population statistics that I have, it's about a 1 in 50 chance. But this is the magic population where we do see a little bit higher recombination rate. So in a biparental cross, I would expect that to be a little bit less. Yes. Yeah, so it's only a few hundred base pairs upstream of, so it's right there in that promoter region, which would kind of lead you to believe that it just happens to stop whatever transcriptional activator that lands there, something, some cis element, and it can no longer function. So the SNP and the indel are 182 base pairs away, so they're right next to each other. And this little indel is just a 3 base pair insertion within a repetitive ATT region. So it doesn't seem like that changes anything, even when you look at transcriptional like analysis, trying to like understand our target sites, like our potential locations. And it seems like it doesn't change anything right there. But the SNP location, that's for some reason, that seems to be our key point. Mm-hmm.
So, like, what do you think to measure?
Yeah, so based on that, it seems to do about what FAD-B does, which is give 20%, let's say, on average. So whenever you have FAD-A and FAD-C, but FAD-B is wild type, you get 60, and then that kind of like all the different combinations of those gives us are different. And that's what we're seeing in that distribution, along with a little bit of like our environmental factors. So that's where that normal distribution is really coming from.
Last question.
So we looked in other locations, and it does have some transcriptional activity in other regions, but this is in TIFF Runner, so we have those locations going. But we're really interested in the seed content because that's where we're seeing our phenotypes at.
Thanks to our speaker. Well, this will be the last presentation of this section. I want to remind you we have the last 4 papers for the those PhD grad student papers. Some of it will be after lunch, beginning at 1:30 in this room, and I do encourage everyone to participate in the flavor following the after this section. For our last presentation of this section, titled Expression Resistance of the Peanut Line, GPN CWS6, the Oleic Capital.
Thank you for that introduction. I guess I should preface this off. There was a mistake there at the time of submitting my article. As you can see from this research here, we are dealing with Mulajan arid area. So just make that clear. I hope that doesn't distract from anything. But anyways, thank you for that introduction. As mentioned, my name is Ethan Foote. I am under the direction of Doctors David Jordan and Jeff Dunn. So as to say, I'm not directly a breeding student, but I sure hope to do this work justice, especially compared to some of these great presentations we just heard. All right, so just as a basic introduction, especially as we look at plant parasitic nematodes in the field, there are many plant parasitic nematode species that can affect many crops, but especially as we look at in peanuts, there's still a handful of significant plant parasitic nematodes affecting peanut. So as we can see from that, our plant parasitic nematodes can highly impact our yield, but also the quality, leading to losses for our growers. They go through, the nematodes can feed on or inside the roots. This can damage the formation of the roots and also lead to damage on the pods, leading to that quality decrease. This damage on the roots can lead to nutrient and water deficiency as movement through the plant is disrupted, and eventually this can lead to plant death. So as we start to look at Mulajan area, which is the peanut root knot nematode, we'll start to look at with the peanut specific is race 1. Race 1 can infect and reproduce on peanuts, while race 2 does not. The symptoms of this is very similar, as you see, it's just normal, or that's the general description for plant parasitic nematodes. However, we can see the characteristic going on our plant over here. Plant or peanut root knot nematode can lead to going on the roots, pegs, and the pods. Again, leading to those misshapen, kind of stubby roots, leading to the nutrient deficiency uptakes as it damages the roots. The range for Mulajan area is prominently in the southern producing regions within the United States. However, as we all know, pest populations can shift and evolve as we go throughout the years. So it's important to stay on top of it, especially from a line development standpoint. As we talk about management of of nematode in peanuts in the field, we can see that our options are relatively limited, especially once populations are developed, they're established in the field. From this, a lot of our approaches are done at the time of planting or prior to planting. And the best way to accomplish this is through an integrated approach. You can look at some cultural practices, which is going through things like sanitation of equipment, going from field to field, field selection, avoiding problem areas in the field, basically just avoidance of the issue. We can also look at crop rotation. This can be accomplished by rotating to a non-host crop, especially if we look at your nematode species of interest. This can help limit population growth in the soil and lower the initial inoculum of interest. We can also look at our chemical application. Again, this is just done at the time of planting and used just as an in furrow application. And again, the modes of actions and the products are limited in this result too. So as you can see, we can get pretty backed into a corner with nematode species in the field and nematode issues. So it's important then to look at cultivar selection, and hopefully if you're in the area or you have resistant cultivars available, it has proven to be a great option for managing peanut nematode in the field. So with that, as we start to look at just host resistance in general, we've all seen over the years that it's important for evaluating pest resistance traits, but then also incorporating those into resistant cultivars is used, as we start to combat evolving and changing pest populations in the field. So with that, we've seen not only in peanut, but multiple species and in multiple disciplines, our host resistance has proven to be an affordable, but also reliable solution to our growers. It can make things simple, make decision-making in the field less complicated and less involved for the farmers, making their job a little bit easier. But we can also see that it can reduce chemical use in the field. And then with that, we can also see a reducing pressure from our chemical resistant development. So one thing to notice with that, is when we look at host resistance, there are actually currently no Virginia market type cultivars available that are resistant to nematode species in the field. So that is something to consider and something to keep into consideration as we continue to develop lines for our VC growers. So if we start to look at resistance to sources of resistance for peanut root knot nematode in peanuts. We have seen plenty of work, extensive work, multiple presentations over this weekend on the region on chromosome 9 from Arachis carnaceae developed. There's been several lines and a lot of work developed from this integration. There's one thing to consider about this integration. It is a pretty large integration. It takes up a large percentage of chromosome 9. So that from a breeding standpoint or a line development standpoint, this can be pretty challenging or undesirable because oftentimes with those large integressions, you see an incorporation of oftentimes undesirable genes with that. So as we look at the resistance on chromosome 9, we see that the majority of the resistance can come from the beginning of the genome. However, work from Dr. Clevenger has shown that some of the resistance is still from the second half or the bottom half of the genome. As we start to look at some of our resistant cultivars available, the options that we have available for developing lines, we have seen that the TIFF-NV line, a runner market type variety, has shown has been able to display high resistance to peanut root knot nematode, and it comes from that large integration from Arachis carnaceae. If we start to look at some of the work in the North Carolina State breeding program, so as we look at our Virginia market type cultivars, we can see some work by Tom Stalker in 2022 that has released a germplasm line, GPNCWS6, that advertised as a root knot nematode resistant genotype. From there, continued work had been done in the last couple of years from Cassie Newman, also in the peanut breeding program at North Carolina State, that has identified a very similar but not totally identical 4 megabase pair region within the genome, as we see on the Arachis carnaceae on chromosome 9. So we started to show some interest in this genome or in this section of the gene to see the performance that we might have and the continued work we can have off of that. So with that, our hypothesis was for our test is that our 4 megabase pair integration in GPNCWS6 will confer resistance to that similar of a larger integration that you'll see on a line such as TIFF-NV IOL. And then our objective goes along with this research, goes along with our hypothesis there, that we were to evaluate the performance of our GPNCW6 line resistance to peanut root knot nematode infection compared to a cultivar such as Bailey 2, which is known to be fully susceptible, and then to the TIFF-NV line, which is shown to be highly resistant. So we conducted work in the greenhouse. We had 2 separate runs with 8 replications in the work. Each of our experimental units was 1 plant. Each of our 3 lines, Bailey 2, TIFF-NV, and the GPNCWS6. From there, these plants were placed and they're planted in a 6-inch pot and separated on the bench, like you can see on that photo on the right there, and able to reduce contamination between the pots, especially as it comes to watering, movement in the greenhouse, things along those lines. So 2 weeks after planting, the plants have been established. We inoculated the plants at either 10,000 eggs per plant or left untreated for each of the cultivars. After 60 days of the peanuts allowing to grow, the nematodes allowing to infect the roots and reproduce, at that time the trial was terminated. The plants were removed from their pots. The foliage was removed from the plants, and the roots were washed clean of the soil. And once the roots were clean, we collected a root weight for that. Once the roots were clean, you can see that we collected a average kind of visual going on the roots. Our rating went from 0 to 100, with 0 equal to no going and 100 to that full going. On the left side there, you can see a nice clean root mass, no average or no going, no obvious going. So that would equate closer to our 0. And then on the right side there, you can see our going at the base of the plant. This does not equate to 100%, but it was kind of on our higher end of the scale. Probably about, this rating was about a 15% going. And then as we move into our egg extraction to count the eggs, we use the Hussey and Barker bleach extraction method, which is a pretty simple process of washing the roots in a 10% bleach solution for about a minute, and going through, washing them off with water, catching them in the sieves, and then collecting them in the sample. From there, we pulled about a 1/4 milliliter to a 1 milliliter sample, depending on the populations in the sample, and placed under the microscope, and populations were counted. From our sample size, we were able to calculate a total final egg count within each sample just by scaling it up to our total solution. Populations would have been so high, it would have been impossible to count all the eggs. So from that, our final egg count, we were able to calculate a reproductive factor. Our reproductive factor was just our final egg count over our initial egg count, and our initial egg count was at 10,000 eggs at a time of inoculation. So as you see, a reproductive factor of 1 or greater would increase or would represent a line that showed that was more susceptible, and a reproductive factor closer to 0 would be of a line that was showed higher resistance. And then from there, we took all the data we collected of our going rating, our total egg counts, our eggs per gram of root, and our reproductive factor, and subjected that to an ANOVA and SAS using a mean separation of Tukey's HSD at a significance level of 0.05. Just as some quick basic results from that we saw across all of our lines, we did not see any going on the pods or pegs, but we also did not see any above ground symptom differences between either our inoculation treatment or our cultivar treatment. From there, I believe that this is mostly probably due to just the time that we allowed for the peanuts and the nematodes to grow in the plant. I believe we would see some more of these symptomology, especially on the later developing plant parts, and more damage to the roots as we left, if we were to leave the plots longer in the greenhouse. But moving on from that, we saw that our root knot nematode inoculation and our cultivar and their interaction were significant for all of our factors that we measured against, being our root going, total egg counts per gram of root, and our reproductive factor. For the sake of time, I'll just go through this real quickly. Obviously, it's clear to see and expected that we saw an increased nematode activity with our inoculated compared to our untreated. And here's just a quick photo to show that this photo is of Bailey 2 on both sides, and on the left we have our untreated, and on the right would be the inoculated. Left side showing nice, clear, healthy roots, and on the right side you see that going again at the base of the plant. There's just a close-up photo of that as well. It's pretty easy to differentiate between the going on the roots and the nodule formation from the bacteria. Again, I'll just kind of flow through this real quickly. Again, our results showed that when pulled over our nematode inoculation and our run, we saw that our Bailey 2 showed increased across all of our factors of root going, reproductive factor, total egg counts, and egg counts per gram of root, which was significantly different compared to our GPNCWS6 and our TIFF-NV. Again, just some visual representation on that. On the left side there, you have the TIFF-NV, in the middle, the GPNCWS6, and then our Bailey 2 on the right. These were all inoculated, and it's clear to see the performance difference between our 2 on the left compared to our Bailey 2 on the right, showing that, again, that increased going at the base of the plant. So here I'll spend a little bit of time on this chart. This is just showing our combination of treatment factors between our cultivar and nematode inoculation, and showing its results or effects on our peanut root knot nematode activity when we pulled over our 2 separate runs. We can see that our only significant difference, and it was consistent across all factors, was our Bailey 2 inoculated, which was significantly higher for all factors than any other combination of either cultivar or nematode inoculation. We can look at our TIFF-NV inoculated and then our GPNCWS6 inoculated on the bottom. You can see similar results between the two, but slightly higher activity of our root knot nematode activity on our GPNCWS6 line. So as we go into our summary, we can just say that these results confirm the prediction of Cassie Newman's work from 2022, that GPNCWS6 will display resistance to that to peanut root knot nematode. We saw that our performance of GPNCW6 was similar to our to the TIFF-NV line. And then even though there are no significant differences noted between the GPNCW6 and the TIFF-NV line, we did see a slightly better performance or a reduced nematode activity in the TIFF-NV line compared to the GPNCWS6. And this is slightly what we expected due to the considerably smaller integration on the GPNCWS6 line. Just to revisit our hypothesis, we can say that we failed to reject our hypothesis that GPNCWS6 will confer resistance comparable to that of the TIFF-NV line, even though it has the smaller integration. And then just to wrap things up, future work is being done now to evaluate the exact gene responsible for resistance in our GPNCWS6 line. And once that gene has been identified, more resistant lines can be easily developed for the Virginia-Carolina region. My references, and then just to thank you for all the supporting staff that I had with this between Dr. Gorney's lab and Dr. Jeff's lab, my committee members, and the support from the North Carolina Peanut Growers Association. Thank you.
I'm sure you'll be happy to answer your questions after the session. That concludes the morning session of the Joe Sugg. Look forward to you joining us this afternoon for the flavors and closing. Thank you. Final section for the Joe Sugg graduate PhD section. To most of you all over 4 scholarships. Reiterate. You got 15 minutes, 12 minutes, I'll stand up, 15 minutes, we'll sit down. So, thank you. This is one of the most important sessions we have. At papers, I appreciate all the presenters, point the effort, don't take a lot of effort. All for attendance. Any later meetings, similar problem. That's it. Hear that, James? We're just talking about age acid injury. I digress. It's been about 20 years ago at the APRIS meeting, plant thought session. A.J. Jackson was in charge, moderator. He looked at the audience and he said, "This ain't our first," his exact words were, "This ain't our first rodeo." And I'm going to let everybody keep up with their own time. 2 hours over. I swear. So, if you ever hear that A.J. Jackson, "This ain't our first rodeo," around the sheriff today, and so we will keep up with that. All right, so beginning our presentations today, our first one is entitled Peanuts versus Respawn, Growing Our Defenses with Wild Species. Alexi Dong will be our first presenter.
Hello, everyone. My name is Alexi Dong, and I'm a PhD student at the University of Georgia in the Department of Plant Pathology. Before I start my presentation, I first want to talk about one of the best plant-themed video games of all time. And that video game is, of course, Plants vs. Zombies. For those of you who don't know, the basic premise of this game is that the zombie apocalypse is upon us, and it is your job to defend yourself against hordes of brain-eating zombies with your botanical arsenal. You're able to deploy this arsenal in your garden, and they shoot projectiles and defeat the zombies. And this is kind of how I like to see my research on peanuts. Peanuts are a huge commodity, and they need to be protected, not from brain-eating zombies, but from other things like diseases. So I'd like to introduce you all to my rendition of Plants vs. Zombies, Peanuts versus Leaf Spot. In this game, we have 2 main characters, 2 main sides. The Peanuts, who are the protagonists, who we want to win. And unfortunately, we have to have the antagonists, the diseases. But first, I'm going to start by talking a little bit about peanuts. So, as I said previously, peanuts are a huge commodity. They are grown globally, and over 50 million tons are produced each year. And the United States is the 4th largest producer. Of that, we produce many different cultivars. And the number 1 cultivar we produce here in the United States is, of course, Georgia 06G. And if we were to look at a description of Georgia 06G, we would see that it is the species Arachis hypogea. In fact, if we were to look at any and all of these cultivars, we would see that they're all the exact same species, Arachis hypogea. And this poses problems because there's not that much disease resistance found naturally in the Arachis hypogea germplasm. And of course, this leads into serious problems when we look at the opposite side, the diseases. There are many different diseases that impact peanut production, such as stem rot, tomato spotted wilt virus, late leaf spot, and of course, my pathogen of choice, early leaf spot. Early leaf spot is a disease caused by the fungal pathogen Passiflora arachidicola. It causes these necrotic lesions to form on the leaves, followed by this chlorotic halo. As the disease develops, these lesions will get larger and larger, and in the necrotic spot, they will actually start to sporulate. These spores, or conidiospores, can result in secondary infections once they are wind or water dispersed. In the field setting, it can cause severe defoliation, and unfortunately, a defoliated peanut cannot produce. And so it can result in up to 50% yield losses when left untreated. How do we defend against this disease? Well, the main way we defend is through the use of fungicides. However, in recent years, there's been a little bit of a lack with efficacy of these fungicides. It was reported in 2021 in Georgia alone that over $40 million were spent for leaf spot management. This is for both early and late leaf spot. And despite these methods, there was still about $31.1 million lost for peanuts in Georgia alone in 2021. So the efficacy of these fungicides are decreasing. In addition to this, the use of fungicides have a potential for a huge environmental impact, especially when used improperly. So what do we do about that? Unfortunately, there's not much in our arsenal right now. However, all hope is not lost because I have heard of a recent rumor of a new character being unlocked in the game. And that new character is wild peanut. It has been shown there's a lot of diversity within wild peanuts, and some of the diversity has a bunch of different traits that are beneficial that can be brought into cultivated lines. And so it is the goal of the Wild Peanut Lab to look at the secondary gene pool of wild species, identify beneficial traits such as disease resistance that can be brought into the primary gene pool of Arachis hypogea to breed a better peanut. However, there is a little bit of a challenge when it comes to breeding these peanuts because in the secondary gene pool, these wild species are diploid. So they either have an AA or a BB genome. In contrast, Arachis hypogea is a tetraploid and has an AA-BB genome. In order to overcome this challenge, the Wild Peanut Lab, we commonly will create synthetic wild allotetraploids. To do this, we get a B genome plant like Arachis valida and cross it with an A genome plant like Arachis microsperma. We cross them together and get a sterile diploid that is then treated with colchicine to induce chromosome doubling. Once this happens, we then get a synthetic allotetraploid, which we will name Valmicro after the parents, and it is now a AA-BB genome that is cross-compatible with cultivated peanut. And so these can be utilized in the future for breeding programs to identify disease resistance and can be brought into the cultivated lines. So for my research, I wanted to look at 13 different wild species that are listed here, some for the B genome, some for the A genome. I specifically wanted to look at these wild species, one, because there's not a lot of research done on these specific species and these accessions, but also because Dr. Mark Hopkins in the University of Georgia Wild Peanut Lab was able to create wild hybrid allotetraploids with these species. He crossed these together and created 8 beautiful allotetraploids, which I was able to use. And so the objective of this experiment was to look at the wild hybrid allotetraploids and the wild species and identify potential new sources of resistance early leaf spot that could be introgressed into cultivated peanut. We hypothesized that there would be a lot of promising resistance within these wild species as they haven't really been researched before. So I grew these plants in a greenhouse setting. You can see some of the wild diploids, the hybrid allotetraploids, and the cultivated controls that I compared them to. For this experiment, I had 3 different cultivated controls, being Georgia 06G, the most commonly cultivated peanut in the United States, Tiff Runner, and Bailey. Bailey is supposed to be one of the more resistant varieties of peanut to early leaf spot. So I utilized that as a control, kind of as a check to see how this bioassay would do. To look for disease resistance, I did a detached leaf bioassay, which looked for phenotypic resistance. A detached leaf bioassay is a relatively simple process in which you grow your plants in a controlled setting like a greenhouse. You then take cuttings of your plants and put them in a moist chamber. For our lab, we commonly use a petri dish with a cotton pad, a piece of filter paper, and a microscope slide, very high tech. We then get a spore suspension of your desired pathogen. For me, it was Bacillus arachidicula. You then harness your artistic ability and paint the spores onto the leaves with a paintbrush. After doing so, you put the lid on the petri dish, put it in the growth chamber, and watch the disease develop. When this disease develops, obviously we're going to see many different symptoms, mainly the lesion development. And so every other day after the first occurrence of lesion development, I would record the number of lesions I saw on each of the genotypes. In addition to this, I would also look at the number of sporulating lesions, as this is very important for secondary infections in a field. So I would use a microscope, a dissecting microscope, or a magnifying glass to look at these leaves and actually look and see if it was sporulating. It kind of looked fuzzy, and so if I saw that, I would count that as a sporulating lesion. So with these two factors, I was able to look at the disease progression over time. In addition to this, the other metric I looked at was disease leaf percentage. At the end of the bioassay, I would take the leaves and scan them and utilize APSSS 2.0 to get the total leaf area as well as the percentage of the disease leaf area, because it's important to know how much necrotic tissue there is because necrotic tissue cannot photosynthesize. For this experiment, I had 24 different genotypes that I tested with 3 controls, and there were 10 replicates of each of these genotypes. And of those 10 replicates, 8 of them were inoculated with a spore suspension of Bacillus arachidicula, and 2 of them were inoculated with just a tween 20 solution. This was done to ensure that there was no disease on the leaves prior to the inoculation due to greenhouse settings. I ran this experiment 3 times with 3 different trials, so there was a lot of work that was put into this. Phenotyping can be a relatively simple process, but it is very time intensive, as I would spend many hours in this room counting lesions. But luckily, I was able to get some really good results, which I'm able to share with you today. First, I want to give you a kind of overview of what these genotypes looked like at the end of the bioassay. So here are some visual representations of some of the leaf scans I took at the end of 20 days. Here you can see in this red box highlighted Tiff Runner, Bailey, and Georgia 06G, our cultivated controls. And they are very susceptible, which was expected because they are Arachis hypogea. What's very interesting, though, was that Bailey has a lot of disease. And it just goes to show how vigorous this bioassay is because it was able to overcome the slight resistance that Bailey has within its germplasm. Similarly, we can see that there were other genotypes that had pretty bad resistance. They were very susceptible in both the wild diploids as well as the wild allotetrapoids. But more excitingly, we can see that there were several genotypes that had very strong resistance. You can visually see they have little to no disease on their leaves. However, we can't just trust visual assessment. We also have to look at the data. So for the different data that I collected for the disease progression, I ran an analysis of variance and then did a Tukey HSD test to look at the statistical differences between each of the genotypes. Here we can see a box and whisker plot showing these disease progression over time compared to the genotypes. At the top, we can see Georgia 06G, Tiff Runner, and Bailey grouping together with a lot of disease, which was expected. We saw that it looked like it had a lot of disease visually, and it's also Arachis hypogea, so it should be susceptible. In contrast, we can see there is a group down here of statistically significant genotypes that have very promising resistance. Some of them mainly include Arachis henni, Magna cum laude, Magna henni 1, and Magdia 1. These genotypes had very little to no disease, which is very promising for future work. If we were to look at disease leaf area or the percentage, we would see a very similar trend to this as well. At the top, we see Tiff Runner, Bailey, and Georgia 06G. And at the bottom, we see some of the familiar genotypes, again, being Magna henni, Arachis henni, Magna cum laude, and Magdia 1. And so to summarize this work, I was able to identify 4 different genotypes that had very promising resistance, one being a wild diploid, which was Arachis henni, and 3 of them being wild allotetrapoids, Magdia 1, Magna henni 1, and Magna cum laude. The implications of this research are huge because they can potentially be used in the future for breeding programs. Future work for this includes potentially using Magdia 1, Magna henni 1, and cum laude, back-crossing them with cultivated lines and creating a segregating F2 population, and then screening those for resistance and finding the genomic region that is controlling the resistance. This can then be utilized in the future for breeding lines so we can get a better peanut. And so, hopefully by the end of all of my work, We will be able to deploy these wild species, these introgressions within our fields so we can defeat the early leaf spot apocalypse. And with that, I would like to thank the UGA Wild Peanut Lab for all of their support, in addition to our supporters for their funding. And of course, I would like to thank all of you for your attention. Yes. Okay, the question is asking, instead of doing the detached leaf bioassay, have I looked at using a whole plant? As of now, I have not done the whole plant. Using the detached leaf bioassay is pretty beneficial for us because we're able to get multiple replicates with very few plants. There's limited greenhouse space, unfortunately. That's just how it is sometimes. In addition to this, it allows some of the canopy structures of these plants are very different. So it'd be kind of hard to compare them together. For the wild species, they're very viney, and for the more cultivated or bushier ones, they would have a more human microenvironment, which could be, more beneficial for the pathogen and just increase the disease, but that might not have to do with the resistance itself. It just has to do with the environment. Yes. Oh, sorry.
Early spot. Do you think there's more variability among wild species that might be able to characterize better? How well has the variability been on within not just disease avoid, but other wild species?
Okay, the question is asking about the variability within wild species for early leaf spot resistance and just resistance in general. I personally found that there seems to be a lot of variety within the different accessions. Some of the accessions will have very strong resistance rather than the other ones, which will have low resistance. So I'm unsure why this is happening, and we'll have to be researched further to understand why this is actually happening. Okay. The question is asking if I have field data for these trials. Unfortunately, I do not have field data because these are wild species. They are not really accustomed to the Georgia weather. They are very viney. And so again, with the microenvironment, it wouldn't really work. However, in the future, once we are able to identify potential introgressions, we can utilize that and then put genotypes that have more of an Arachis hypogea background into the field to verify that this resistance is going to work in the field.
That's the time you have the questions right now. Thanks for listening. We'll be earlier that I've got the easy job and Dr. DeWard does all the hard work. Yes, that's true. That's true. All I have to do is speak. Yes, just really go navigate. All right, our next presentation this afternoon is entitled Evaluating the Potential of Inoculation with ACC deaminase containing rhizobia to improve peanut drought tolerance. And Joan John is our presenter this afternoon.
Thank you for the introduction. Hello, everyone. I'm Joan Chung. I'm a PhD student from Auburn University working on peanut drought tolerance. So today I will talk about one of my projects entitled Evaluate the Potential of Inoculation with ACC deaminase rhizobia to improve peanut drought tolerance. So as we know, drought is one of the biggest abiotic stress that causing yield reduction. And in 2011, the United States experienced its worst drought. So on the map on the top left corner is showing around 88% peanut field are suffering from drought in August 2011. So this timing is particularly important because in August, the peanut undergoes the pod filling stage. So that is when the water demand is really high. So therefore, the mid-season drought has a devastating effect, can causing yield reduction for peanut of up to 85%. And in Alabama, we have the high temperature in the summer, And also the drought is often related to the rain event. Oftentimes there's no rain for a few weeks, and also the sandy soil in peanut field can make the drought worse. And also in Alabama, there is 90% of peanut field are not irrigated because irrigation is expensive. It costs around $200 per acre per year to run it. There is a picture in the middle showing, I took this picture from my rain-fed field in Hellland. So we can see the leaf, they are curling, and also they feel like crispy in the rain-fed condition. But we know breeding for drought-tolerant cultivars for peanuts have made a significant progress, but we know it's a very time-consuming and costly process. So here, we're trying to look for alternatives like inoculation, so they are faster and more economical and broadly applicable. So in this research, we're trying to explore how the ACC deaminase rhizobia can improve the peanut drought tolerance by altering the ethylene production pathways. So in the plant, there is a hormone called ethylene. Most of you probably know it by the fruit ripening, used for banana ripening the banana and other fruits. And the ACC is the immediate precursor for ethylene. So without any abiotic stress, so the ethylene plays a very important role in regulating the seeds germination and the root and leaf growth and nodule formation is for legumes. But when the drought strikes the plant, the ethylene production increases by 2 peaks. So the first small peak is produced by the plant, transforming the small amount of ACC that is normally present in the plant into ethylene, which triggers some genes related to different plant defense mechanisms. And later, after a few days of the first peak, the second peak occurs. So the plant produces additional ACC. So in response to the abiotic stress, like drought. So the second ethylene peak is much larger than the first one. And also it has a very bad effect such as leaf loss, leaf yellowing, and growth inhibition, and also physiological like trace reduction like photosynthesis and stomatoconductance under the stress. And but we know like plants, they're not stupid, they're really smart, they know how to look for helps. There are some rhizobia in the soil that they contain a very special enzyme called the ACC deaminase, which is encoded by ACDS genes. So this enzyme can break down the ACC into ammonia and alpha ketobutyrate. So this ammonia and alpha ketobutyrate providing energies for rhizobium in the soil, but also reduce the magnitude of the secondary ethylene production by around 50 to 90%. So this reduction of ethylene level can prevent or reduce the negative effect of the plant stress response during the secondary ethylene peak. So therefore we think, oh, maybe when the plants inoculate with this ACC deaminase rhizobia, they can help improve the peanut drought resistance. So in this experiment, we planted one cultivar called AP3, which is a drought-sensitive cultivar in the greenhouse between 2021 and 2022. So for the water treatment, we have the drought treatment. So we let the water holding capacity gradually reduce 10%. And in 2021, we had a short drought. In 2022, we have a prolonged drought. We have a well water treatment, and that is we maintained the water holding capacity 100%. And there were 4 types of inoculation treatment, including no inoculation as a control, and wild type inoculation with Bradyrhizobia species strain 9, and 5 times plus inoculation with ACDS gene overexpressed mutation strain, and the mutant negative inoculation with ACDS genes deficient mutation strains. And so we tested those mutant strains and the wild type strains about their ACC-DM and its activities in the lab. And the data from the lab we tested showing there is a significant difference on the ACC-DM and its activities. We applied the randomized complete block design for this experiment with 4 reps in 2021, 8 reps in 2022. We took multiple measurements for photosynthesis, monoconductance, SPAD during the drought. And the picture on the right side is showing me taking photosynthesis, monoconductance measurements using the LICOR 6800. And at harvest, we collect the data for leaf water content, biomass for pod, root, shoot, also harvest index, carbon isotope discrimination, and the delta nitrogen 15 and the nitrogen derived from the atmosphere. So it comes to the result. We did the principal component analysis. It tells us that PC1 and PC2 explained around 75 to 57% of the phenotypic variance in 2021 and 2022 respectively. And also the PCA separate very well between 2 water treatment. So we can see in the well water treatment plants, they were located on the top side of the plot in 2021, and then the drought treated were on the other side of the plot, similar to 2022. And if we look closer, so we can notice the uninoculated and the uninoculated plants were well separated with the 5' plus and the wild type, no matter under what water condition. So we can see that in both years. However, the mutant negative overlap the uninoculated and the wild type and 5 times. And we have seen that in both water conditions and in both years. And in terms of the correlation, and we see that the biomass for root, shoot, and pod, and nitrogen percentage, and nitrogen derived from the air, they are highly positively correlated with each other. And the delta nitrogen 15 is negatively correlated with them because the less delta nitrogen 15 value means the plant is doing more biological nitrogen fixation. So that's why it's negatively correlated with the nitrogen parameters. And also we can see the photosynthesis, the monoconductance, carbon isotope discrimination, leaf water content, they are positively correlated with each other. We're seeing that in both years. So in terms of the biomass, in this plot, the solid bar means they're drought stressed, and the bars with the pattern is well water treatment. So the first thing we notice is the biomass for shoot and root in 2021 is much less than 2022. This is because in 2021, the plants had less days of growth than 2022. And also this result is telling us, oh, the drought is not significant, reducing the shoot biomass in 2021, and it didn't have a significant effect on the root biomass in both years, neither. But the drought significantly reduced the shoot biomass in 2022. We can see the difference under 2 different water treatments. For the inoculation treatment, we didn't see significant effect on the shoot biomass in both years, but we did see significant difference in the root biomass. So the plants that didn't have any inoculation, those controls had significantly less root biomass compared to the plant inoculated with strains, including the mutant active, wild type, and 5 times. And similarly, we see that in 2022. And also we notice there is an increased trend in the shoot and the root biomass in 2021 that the plant inoculated with mutant negative. But we didn't see it in 2022, and we're still trying to understand, oh, why this is happening. So we measured multiple times for photosynthesis and stomata conductance. We averaged the value. So the result is showing that the drought did significantly reduce the photosynthesis and the stomata conductance. But we didn't see any significant effect from the inoculation. So that means it doesn't, the drought reduced the photosynthesis stomata conductance no matter what inoculation treatment there was. Later, we calculated the nitrogen amount derived from the air, and it's calculated by multiplying biomass of the plant tissue with the nitrogen derived from the air. So this special thing for this experiment is to calculate the nitrogen derived from the air, we obtain our own B value by planting the peanut in the pot in our greenhouse without providing any nitrogen source. So this result is telling us the drought does not have significant effect on the nitrogen amount derived from the air in the shoot in 2021. And also it didn't show significant effect on the root biomass, on the nitrogen amount derived from the air in the root in both years. But it did show significant difference in the nitrogen amount derived from the air in the shoot in 2022. Luckily, we are seeing significant difference in the inoculation treatment in both years in both parameters. Again, we're seeing the plant that didn't have any inoculation have significant less nitrogen amount derived from the air in both root and shoot. So there is no significant difference among the mutant negative wild type and 5 times in the nitrogen amount derived from the air. And once again, we see that increased trend for the plant under drought condition inoculated with mutant negative. They're showing an increase on the nitrogen amount derived from the air in the shoot. So with that, we conclude the drought effect is significant on the plant growth regardless the inoculation type. And we didn't see the effect of ACC deaminase rhizobia mutant on drought tolerance. And the absence of effect of modifying the ACDS gene expression could due to short and intense drought. And the ACDS mutations doesn't affect the nitrogen amount derived from the air, photosynthesis stomata conductance, but it does affect the root dry biomass, especially inoculated with mutant negative. So thank you for listening, and I would like to take some questions.
Time for a question. My question is, from the first year to the second year, you had a short drought in the first year and then changed to a long drought. Why was that part of your methodology, and how does that affect your interpretation of results?
So the question is, in the first year of my experiment, I have a short drought. And in the second year of my experiment, I have a prolonged drought. Why was that? So the reason we said it's not originally planned like that, but in the first year, we had a 1-week drought. And at the end of 1 week, the plants just crashed. So we only can maintain like for 1 week. And in the second year, the weather is really drought, really hot, and the transpiration is really big. So the plant crashed in 3 days. So we're only able to take the photosynthesis or physiological trait measurements for only 1 or 2 times. So that's just not enough for us to understand what's going on. So we fully recovered the plants. And then later we carefully maintained it to gradually reduce the water holding capacity for another week so we can take more measurements about it. Yeah.
Thank you. Yes, it's time we got any questions. Thank you. Our next presentation is entitled Ramp Response to Delayed Timings of Four-Zone Break and Trilox Oxygen Ribosol. Yeah, that. I think we can all agree. All right. And our center is next.
Put it down. All right, just making sure it works. All right, thank you, Dr. Kemmerite, and good afternoon, everybody. Today, I'd like to share with you some of the findings of our research looking at peanut response to the delayed timings of these two herbicides. And so often it is quite difficult to get new herbicides registered for use in peanut production. However, Brake Fluoridone was registered in 2023 as a new mode of action and welcome addition to help target some of the many problematic weeds that are typically targeted with other pre-emergent herbicides such as Valor. And so you may be wondering, well, if we already have a racehorse in the stable, why do we need to continue to add new herbicides? And so here I wanted to present my case for additional herbicides. So as many of you have probably heard multiple times throughout this talk this week, is that U.S. production, across all the U.S. production, 94% utilize at least one herbicide application throughout the growing season. And 62% utilize Valor as a pre-emergent application, flumioxazin. And more specifically for us in Georgia, over 74% utilize flumioxazin as a pre-emergent herbicide. And so you can see how this can be concerning when we're using repeated use of the same mode of action over and over again across not only peanut, but other production systems as well. And this ultimately will lead to increases in herbicide resistance. And so Dr. Randall Singleton et al. At the University of Georgia found and confirmed just that in recent years. This Georgia Palmer amaranth population was resistant to PPO inhibiting herbicides, including Valor. And so this study found that Valor applied at a 3x rate. This population was 31 times more susceptible, or was 31 times more resistant than the susceptible population. And so you can see the importance of diversifying our weed management portfolio and adding in new modes of action, which for us with this herbicide Brake is a phytoene desaturase inhibitor. And what that is a bleaching herbicide, which typically turns the plant tissue white. And so as a new herbicide to work with, we're always trying to investigate what the limitations may be with this herbicide. And so currently the label states that this herbicide is to be applied behind the planter or within 36 hours after planting. And Dr. Kemmerite, Dr. Ebney, I'm sure you always get a phone call from the growers in the state of Georgia that tell you, I got all my applications out timely this year. Thank you for all that you do. But most of the times, oftentimes they say, I was able to plant, but I wasn't able to get out into the field. And so there's many reasons why that occurs. As you can see here, it could be weather, equipment failures, operational labor constraints, the amount of acres that we're dealing with, as well as the concern for increasing injury, leading to yield loss with this delayed herbicide applications, as Johnson et al. Was able to find specifically with Valor. And so in addition to Fluoridone, Brake, University of Georgia wheat team is always looking to evaluate new herbicides for their potential use in peanut production. And so trifludimoxazin, BASF product, Rexivor, we've been evaluating it as a herbicide, but now looking at it for these delayed applications as well. And so the overall objective is to evaluate peanuts' response to delayed timings of fluoridone and trifludimoxazin, specifically beyond that 36 hours. And so the null hypothesis for this study is that the delayed applications, regardless of timing, will have no effect on peanut density, growth, and yield. And so this study took place between 2022 and 2023 at the UGA Ponder Farm in Tide, Georgia. Cultivar used was Georgia 06G, and this was set up as a randomized complete block design with 4 replications with a 3x4 factorial arrangement. And so factor A included our herbicide, so that's a non-treated control, our fluoridone at a 1x rate, trifludimoxazin at a 1x rate, and then factor B being our timing at 1, 3, 5, and 7 days after planting. And so this data was analyzed in SAS 9.4 with Procglimics, and then utilizing Tukey HSD for our pairwise comparison. And so at each application, one thing we wanted to do was we wanted to evaluate and observe and monitor the development and growth of that seed when we applied it. And so as you can see here, we have what that looked like for 2022, our 1, 3, 5, and 7. And what I wanted to point out here is in 2022, we had greater than 50% ground cracking at 7 days after planting, with roughly 20 to 25% green plant tissue exposed. However, when we look at 2023, when we are looking at that 7 days after planting, we can see some slight differences where we have less than 10% green tissue exposed, which is a cause of common environmental differences between one year and the other. This year it was a little bit colder, so it took a little bit longer. So keep that in mind, as that could influence recommendations for delayed applications. And so common response variables that we look at when evaluating new herbicides include density, stunning, herbicide symptomology, height, width, and yield. And for this presentation, I'll just be focusing on density, stunning, height, and yield. And so there are three main phases when evaluating new herbicides that we're looking at. And obviously the first phase is a delayed application or any application of a herbicide going to impact stand of peanut or the density. And so at 13 days after planting, we observed that there was no herbicide by timing interaction. And so we combined those timings within each herbicide. And so as you can see here, I will set up this graph, and all following graphs will be pretty similar to this. We'll have a response variable on the y-axis and then our herbicide treatment on the x-axis. And so this is density in plants per 1.5 meter per twin row. As you can see, combined across those timings, fluridone did decrease plant density by 6% from the non-treated, but was no different than trifludimoxazin. And keep in mind here is that the threshold that would be of concern is 19. So you can see that we're still above that threshold. So the second phase after we have established that is, how does it impact, these delayed applications impact the growth and development of the plant throughout the growing season? And so looking at 13 days after planting for stunning, we can see that regardless of the timing, they all increased stunning compared to the non-treated control, ranging from 5 up to 11% stunning. And so as another thing to note here is that as we delayed that application, we saw an correlated increase with the stunting percentage aside from 3 days after planting. And so the same trend we saw in trifludimoxazin as well, with ranging between 13 to 19% stunning in that 7 days, again, being the most injurious and the highest stunning percentage at 19%. And so here is just capturing what that looked like at 13 days after planting across those 4 delayed applications. We can see the common symptomology, the yellowing, the bleaching, and the stunning when we are comparing it to the non-treated control for fluridone. And same thing for trifludimoxazin. You can see that bronzing on the leaf tissue, which is very common to other PPO herbicides such as Valor, and the stunning as well when we are comparing it to that non-treated control. And so moving along a little bit later into the growing season, we can see at 30 days after planting, evaluating height, that fluridone by this point in time had no impact on height, whereas trifludimoxazin did decrease heights by 6%. And again, one thing to note here is there was no timing effect, and so those were combined across those herbicide treatments. And here at 30 days after planting, just wanted to capture to illustrate what those peanut plants look like. You can see a little bit of yellowing still occurring at the 1 day and the 7 days after planting for fluridone. And again, trifludimoxazin is probably a little bit harder to see in these photos, the bronzing that's occurring. And you may see that 7 days after planting, they look a little bit more skippy than the non-treated control, but it's really hard to tell, especially at just 6%. So the last evaluation of our phase 2 was looking at peanut heights at 80 days after planting. And so by this point in time, you can see that there was no herbicide nor timing effect in regards to height, with an average of 38 centimeters across all of those treatments. And so the last phase of evaluation here is looking at peanut yield. And so again, you can see by this point in time, following a similar trend with the heights at 80 days after planting, the peanuts were able to tolerate those delayed applications and grow out of any early season injury. And so we have a range of yield from 53.89 to 54.88 across those treatments, kilograms per hectare. And so in conclusion, here we can see that fluridone did slightly decrease our peanut density with those delayed applications across those timings. But as I mentioned earlier, it was still above that threshold of 19. And so as we moved into the growing season, we did see some common visual injury and symptomology, but that also is an indicator that we had good activation for weed control during the growing season across those delayed applications. And so by the time we got to 30 days after planting, we saw that fluridone had no effect, whereas trifludiamoxazin did decrease heights by 6%. But again, by the time we got to 80 days after planting, we see that there was no effect regardless of the herbicide or the timing. Again, just reiterating that these peanuts were able to tolerate those delayed applications and grow out of that. And so finally, there was no effect on yield, which is the most important component here. So again, the overall takeaway is that these peanuts were tolerant to these delayed applications. And so this gives growers the confidence to know that if there was circumstances that presented themselves where there was a delay to occur beyond that 36-hour window, that we're still going to be able to achieve our yield goal. And so this research supports previous research found by Carol Johnson et al., which looked at delayed applications of flumioxazin on peanut, where as we delay applications, we do see an increase in injury. But the most important thing is that we did not impact final yield. And so this research will be repeated in 2024 for a third and final year. And with that, I would like to acknowledge Seed Pro Ag for their contributions, as well as the many people listed here, Georgia Weed Science and Crop and Soil Science for their support throughout this research. Thank you. Yes, sir. The question was, does the delayed application?
Just using these products.
Yeah. So the question was just reconfirming the delayed applications and how that relates to canopy closure.
Yeah.
Sure. Well, by the time, so as we saw from the photo, when we're looking at 7 days after planting, you saw that there was only about 10 to 15 or 20% of green tissue exposed at that last application. And so we're still able to get good coverage on the ground for the opportunity to get activation with a rainfall event for proper weed control.
Good question.
Yes, sir.
How would you suspect valid before? I know you mentioned that you saw that the previous research had shown that valid kind of showed similar results with the delayed application, but how would you think that would compare to Florida or the other products?
Yeah, so the question was comparing Break to Valor and the contrasting differences. And so I think that a lot of work has already been done with Valor, and if we had the space, it would have been nice to include Valor in there to do a comparison. But there's so much data already out there indicating when to put out Valor. But as a new product, that information isn't there yet for Break. And so that was our goal, was to see what the limitations are of this new herbicide and if those delayed applications would implicate, growth or final yield. Yes, sir.
Would you recommend a label change based on the results?
So the question was, would I recommend a label change? That's a good question. I would say that based on this data, I think we need a little bit more to understand the impact that it can have. I think 7 days after planting, depending on where the growth is, there may be a little too much green tissue exposed at that point. We're doing some other studies looking at post applications and its impact. And so if the ground is cracking, but there's no green material exposed, I think is good. So 7 days may be a little too extreme, but it just depends on the season and how much material may be exposed at that time. And so I leave that up to CPro Ag to decide on whether they want to change their label.
That's the time we got. Thank you. Final presentation of our Sugg graduate student PhD category 2024 English meeting is entitled Magnitude of Fungicide Resistance, Agroadelia rossi, Georgia peanut fields, and its phenotypic and in-plant effects. Jessica Bell.
Well, thank you for the introduction. As you said, I'm Jessica Bell at UGA under Dr. Tim Brenneman, and I'll be telling you about the magnitude of fungicide resistance in Agroadelia rossi from Georgia peanut fields and looking at its phenotypic and in-planta effects. So Agroadelia rossi is the causal agent of peanut stem rot disease and is commonly referred to as white mold, and you can see why from this image. This is a soil-borne fungal pathogen, and it can form sclerotia for long-term survival. This white mycelium can also go completely underground and rot pods without even causing above-ground symptoms. So my first point is that it's difficult to manage. My second point is that it's vital to manage because it can cause up to an 80% yield loss. Now in Georgia, we primarily rely on fungicide applications to suppress this disease, and we have to very heavily apply them, and this has been the case now for 30 years. So anytime you apply a product this long, you obviously have issues of fungicide resistance potentially arising. We use the SDHI, DMI, and QOI fungicides, and we do have some products that are actually very efficacious on this disease. But nonetheless, the justification behind my research is that over the past few years, UGA has gotten a lot of calls from growers across the state of Georgia saying, hey, my products didn't really seem to work for me very well this year, and we would like to know why. And to give a practical example of this regarding last year's peanut production, I actually have a quote from Dr. Barry Tillman. He said that some fields in the western Florida panhandle exhibited an 80 to 90% incidence of white mold, and as you can imagine, these fields suffered some major yield losses. So if we have products that work, like I said, why are we seeing these issues? It could be a variety of things, such as misapplication of the fungicides, or even the fact that we plant a lot of O6G, which is really susceptible. There's a variety of things it could be, but the number one problem is always trying to get the fungicide through the very dense peanut canopy to the soil level, because you can see that's where the fungus is. Now I have fungicide resistance with a question mark, and my project has actually shown that over the past 25 years, we have seen a shift in fungicide resistance when it comes to flutolenil and tebuconazole, which are DMI and SDHI products. Now I'm not going to get into that today, but I do want to explore, or my project was looking into whether or not that shift is causing an efficacy issue as well. Now, before I get to the meat of my presentation, I do want to kind of set up what I've previously done because it'll make today's talk more relevant. I began by collecting 256 isolates from 8 counties across the state of Georgia, and I assessed the in vitro sensitivity of each one using just one dose of 4 different fungicides that I'm going to talk about today. And that's tebuconazole, flutolenil, methyltrifluconazole, and benzophenoflupyr. Now, what that assay allowed me to do was identify my 5 most highly sensitive isolates and also my 5 least sensitive to each of these 4 fungicides. And at that point, I quantified their EC50 values. And I'm just going to very briefly go over those results with you. These are the EC50s in part per million for my DMIs in black and SDHIs in red. And there's two main things I'd like to show. If you look at the least sensitive average, for each 4 of these compounds, they're always a little bit higher than the most sensitive. But with the DMIs, there's not any significant difference, but the SDHIs do show a statistical difference there. My second point is that though we see differences, the difference between the most and least sensitive EC50 values is very small. But nonetheless, we do have differences, so we kind of wanted to investigate that just a little bit. So my hypothesis with those EC50 values in mind is that the most and the least sensitive isolates are probably phenotypically very similar and do not cause different levels of disease in a field when sprayed. So my first objective was to characterize the attributes of these different isolates by looking at the virulence on peanut stems, their growth rate, and sclerotial production. So we'll start with my stem assay. I collected Georgia 12-line main stems from a field in Tipton, Georgia, and cut them in 12-centimeter sections to where the third node was always 3 centimeters from the bottom. I put them in boxes and filled it with sterile sand and half a liter of sterile water up to that third node. I then inoculated at that third node using actively growing cultures, and I incubated these boxes in the dark for 5 days at 25 Celsius. And this was a randomized complete block design with 5 reps. On day 5, this is an example of what I would have seen for my most and least sensitive glutathione isolates. And then I measured the mycelial growth on the stems and the length of bleaching due to the oxalic acid. My next assay was looking at these same isolates' growth rates. So I plated these on quarter strength PDA that was not amended with fungicides. I grew them for 3 days in the same conditions. And then I measured the diameter of those colonies. Now, at the end of this test, I put those same plates back in the incubator, and I left them for 3 additional weeks so they would form sclerotia. I then counted sclerotia and analyzed, this was a randomized complete block design with 5 reps. I analyzed my data in R using an unpaired Welch's t-test on my most and least sensitive groups, and I did duplicate all assays. So for my results, I'm going to show you a variety of graphs for 2 objectives today. And to keep things a little bit simple, I'm going to show all of my most sensitive isolates in the blue bars, and the red will be my very least sensitive isolates. And I'll also be showing 95% confidence intervals. So on the left-hand side, the XI stems is measuring the stem coverage in centimeters. The growth rate is showing you the colony diameter in millimeters, and the number of sclerotia per plate on the very right-hand side. And you can easily see for TEB that we don't see any differences across these different attributes. For methylfluconazole, we do start to see some differences, but again, because those EC50 values weren't even different for these 2 groups, and since the differences are really small, I would imagine that there's not really any biological meaning behind these differences. Now, moving into the SDHIs, if you recall, the least sensitive EC50 value was a little bit higher than the most. So it's possible that what we're seeing on these right 2 graphs with sclerotia and growth rate could be a result of having some resistant mutations starting to arise, which tends to make those isolates a little bit less vigorous, which we call fitness cost. But if that's happening, it's very minor. Now, the biggest difference that we see with benzophenylflupyr, or all the attributes for all fungicides, is with this compound with the number of sclerotia produced. And again, that could easily be a fitness cost, as could the growth rate. And I do want to point out one interesting thing with this. If you look at the growth rate, least sensitive, they do grow the slowest. And again, that's on the PDA plates. But if you actually put these isolates on these stems, the plant material, it's reversed. They grow the quickest. And I think this is a really good example of while in vitro assays are important, they don't always translate to a field setting. So to make my research that I just did a little bit more practical for a grower, my second objective was to try to figure out if those differences that I just showed you in the lab affect disease control in the field. So basically, do my least sensitive isolates cause more disease than my least? So for this, I used the microplots at Tifton, Georgia. There are 1 meter diameter rings in the ground, each containing 3 peanuts, and they're all 3 treated the same way in one plot. I inoculated the base of the main stems using one of my 2 most or 2 least sensitive isolates, and I did so around early August or late July. So every plot that was inoculated, it has one that was sprayed and also one that was unsprayed. So if it received a fungicide application, it was either Aureus, Convoy, Provosol, or Provia, which is tebuconazole, flutalonyl, methyltrifluconazole, and benzophenoflupyr respectively. And just as a little footnote, a Provia is not labeled on peanut, but unlike Elatus, it is just the active ingredient benzophenoflupyr, which is what I was wanting to test. That's why I applied that product instead. So I applied the sprays right after inoculations, and then again a month later, and we did our best to irrigate after each time, just to try to wash off any fungicide deposition. This was a nested randomized complete block design with a 2x2 factorial, where I had 4 blocks, and within each block there were 2 treatment replicates. Upon maturity, I hand pulled the plants and rated them on a scale of 0 to 10, with 10 being dead. I did 3 years worth of this study and used R to run an ANOVA and a Tukey test. All right, so moving into my results, we'll start first with the SDHIs. And on the y-axis, I'm showing you the disease scale. Again, 10 is dead, 0 is no disease. The color scheme is the same as before, where the most to least sensitive are blue and red respectively. This time, the shaded bars were the ones that received fungicides, and the ones that are not shaded did not get sprayed. The 2 main things that I want to show with the SDHIs is first, whenever sprayed, disease was always significantly reduced. So we do see a substantial response to the fungicides. The second thing is if you look at just the spray plots, there's no difference in disease levels, which is what we hypothesized. So they're working well and they're suppressing the least sensitive just as well as the most sensitive. Now with the DMIs, we actually don't see any differences here, which I'll explain in just a moment, but we still see that when sprayed, disease is the same. So moving into my conclusions, for the DMI phenotyping assays, again, all differences were very minor. And I think that since the EC50s weren't different hardly at all, that what we're just seeing is variation due to a small number of isolates analyzed. For the field assay, again, neither responded to fungicide applications. And I have several hypotheses, but one of them is that with the nature of the DMIs, they tend to be a little bit extra systemic. And the irrigation system in the microplots on its very best day was very sporadic. And we think that perhaps we just didn't wash it off either soon enough or well enough to actually get it to the active site. So that's one hypothesis there. Now, the SDHI phenotyping assays, overall, pretty much all differences were similar with the exception of the sclerotia graph. And again, that could be because of resistant mutations. Now, if we're starting to see mutations, one explanation for what we see on the bottom is that sometimes we don't actually see a fitness cost. It actually makes those isolates just a little bit more vigorous. So that's another explanation. For the field assays, what we're saying with these SDHIs is that they're still very effective against peanut stem rot because A, they did suppress disease significantly, and B, they did so to the same extent. So going back to my main point of why fungicides may not control stem rot. As far as fungicide resistance, we're not saying that that's an issue at this time. We're actually not even using the word resistance at all. Instead, we're saying that we have a shift in sensitivity, which I have proven with my data, but so far it doesn't really seem to be affecting disease control in the field setting, which of course is where it matters. So that takes me back to my #1 issue of trying to minimize foliar deposition in that dense peanut canopy. So in Georgia, we have a lot of dry land fields. So we don't have the luxury oftentimes of irrigating after a spray, which is one way to mitigate that. But the best way if you have dry land fields is to spray at night whenever your leaves are closed. And as far as future research, the fungicide resistance aspect of this project is coming to an end. But as with any pathogen, we'll need to continue to monitor for fungicide resistance. And thank you for everyone involved, and a special thank you to Dr. Brenneman for all of your guidance and abundance of patience. And with that, I'll take any questions.
We have plenty of time for questions.
Yes, sir. The fungal load in the soil before planting? We did not. To my knowledge, we haven't done white mold assays in that field or those locations in a really long time, so I would imagine that it would be very, very low at the most. Now, I did have control plants as well, and they didn't have any white mold, so I don't think that would have been an issue. Yes, sir.
Do you have an idea of the fitness cost? Why then?
Can you repeat the question?
So, with you have fitness costs with phobia?
Well, I'm saying that we potentially have a fitness cost. I'm not sure that we do. I would need to look at the genetics behind that.
Okay.
It could just be because of variation with the data, which is also part of what I think it is.
Other questions? Are there examples, Jessica, have you seen examples? I know there's been speculation, but are there examples of resistance developing in other crops or other systems to agrophilic offset?
Off the top of my head, no. I'm not aware.
I know it's a pathogen that affects a lot of crops and a lot of different parts.
Right.
Any other questions? Let's thank our speaker.
Thank you.
Before doing so, right paper competition, I hope you'll agree that it's some of the best presentation certainly some of the most anticipated. I would remind you that at 3:15 today, our poster presenters will also be there, which includes 11 PhD students and 10 master students who will be very happy to talk to you about their research as well. Please support them. That's from 3:15 to 5 o'clock. We follow that with our business meeting and follow that with our assessment. So thanks for being here today, and we look forward to seeing you as we continue the final round